Thank you, dear colleagues, who survived in this evening. I'd like to present our study to you, genomic features of oil model compounds, biodegradation by Rhodococcus strain. There are well-known some pollution types of the soil, for example, heavy metals, plastic, oil and its components. One of approaches to solve this problem is use bioremediation bacteria who can digest complex substances such as alkanes, polycyclic aromatic hydrocarbons, and disrupt all films, remove it from the surface of the soil for the roots, and reduce toxic effect. Now, this approach illustrated by the picture on the slide. We isolated a strain of Rhodococcus genus from the soils of oil-polluted ground or soil near Irkutsk, and this strain can help plants to survive with oil pollution. It synthesizes some phytohormones and digest oil film from the roots. We conducted some experiments with some of the species of plants. For example, we treated the seeds of maize, and as you can see, some properties of this plant: germination percentage, aerial part percentage, and length of the root and mass. All conditions of these samples was equal except for presence of bacteria and oil pollution, about 2% in the sand. As you can see, the presence of bacteria neglect the negative effect of the oil almost completely in some cases. We carry out a lot of such experiments, but I show only two on these slides for maize and pea. The results are very promising. Then we tried to measure an effect of digestion the oil in. Pardon. This is illustration from the roots, the microphotograph of Redka maslichная, Aphanos sativus. As you can see, the presence of the Rhodococcus strain helps to develop the root. As talking about a pure oil digestion and degradation, the number of samples was used, and the best of them are presented on the image. It's very clear that oil film is degrading, and we selected this sample 108 for whole genome sequencing that performed a year ago. We sequenced it with Illumina Novasec and with a pretty good coverage, I think. Assembled with spades and then scaffolded in the whole chromosome with true regout, which I like very much, thanks to Mikhail Kolmogorov from Genome Research paper. This genome, among 400 genomes that are already published in NCBI, this pretty diverse family, consists of 5,700 genes. The details who interested are already published in paper in April. Then we built a phylogenetic tree to found a place in lineage, but it's not very clear here. We determined species, but I don't read it very clearly. It's very funny. Qingshen. And looking deeper into genome for genes genes of interest. We found 18 biosynthetic gene clusters here. Some of them related to antibiotic synthesis. A lot of them, pyrimidine, monocyne, and others. We plan to experimentally verify this synthesis by the cell culture. Some genes are related to adaptation. For example, salt tolerance as ectoine and iron acquisition from the environment, acidophores, well-known enzymes. And coming back to the digestion and oil degrading, we saw that our strain metabolizing some of the most difficult substrates due to its high diversity of different types of mono and dioxygenases. For example, for alkanes and biphenyl, we tried to group these genes to the pathways. Pathways were taken from KEGG, Kyoto Encyclopedia, and for example, very promising biphenyl degradation catabolic pathway. We found some key genes. You can ask me where the other genes. It's a problem with annotation because they are automatically, and most of the gene must be annotated incorrectly and need manual verification and manual search via BLAST or HMM profiling. Alkanes degradation also presented by some genes for LKB, LKM. It's a key genes according to literature. We want to verify it. Our strain, it's not shown on the slides, can grow on pure naphthalene as a model compound. But previously we published a paper, pure chemical research of possible pathways of degrading the complex organic compounds by this strain in 2016. Also, we found some related genes with degrading the polycyclic aromatic compounds on this slide, and we in the start of genomic research of this strain, and in perspective we want to manually verify all the steps of known pathways that are described for Rhodococcus strain, because actinomycetes are well known for degrading the aromatic compounds. And we found some promising databases, BioServe-DB and Remediation-DB last year. They must help us to find the mechanism of degrading that was already studied experimentally. And I'm glad to invite all of us to the upcoming conference in March in Lake Baikal that related to this topic, mechanism of adaptation of microorganisms and to different environmental conditions. The site is still hold the old conference, but it will be the same. In autumn, we already announced it officially, but you are invited right now. Thank you for your patience, further questions, and your attention.
Okay, thank you for your presentation. Could you please show us the slides with tables?
This?
Yes, this one.
Yeah.
The one before. Yes, with Maize. I just saw an interesting thing. You can see here that with bacteria, the mass was lower when you had it oil with bacteria.
So bacteria is better than without bacteria.
If you look at mass in grams, yes. When it was just the bacteria, the mass is lower when you had bacteria with oil.
Yeah, I know the last column.
And it seems like the bacteria were harmful.
It's need to further study, I think, because there are maybe some error in the experiment or no repeating. Not
one repeat.
I don't know if talking fair, maybe 3 repeating only.
Okay. Well, yeah, perhaps with some more repeats it would balance out.
I want to address that this microorganism has different properties. It can synthesize phytohormones, so it has effect on plant even without oil.
Okay. Okay. Thank you.
Any questions? Thanks a lot.